Good morning. In this 12th class biology, we are going to study the next topic, that is outbreeding devices. Outbreeding devices are those devices which are used by the monoecious plants to prevent the cell pollination. Why these ways are created by these monoecious plants to prevent the cell pollination? Because we know that in the cell pollination, the pollen grains of the same flower falls over the stigma of the same flower. So male gamete and the female gamete of the same flower. So the genotype of the same flower. So there is not any other variation, any other trait, dominant or recessive trait, come inside the offspring from any other plant, from any other type of genotype. In this way, continuous self-pollination, continuous self-fertilization creates only the clone organisms. And if the environmental condition, if the environmental factors, biotic, abiotic factors, physical or geographical factors, chemical factors are changing continuously, so this type of plants cannot adjust themselves and cannot have the better survival and fitness. In this way, plants use and create such type of outbreeding devices to prevent the cell pollination. Out of this outbreeding devices, the first is that if the bisexual flower is there, then this synchronization of the pollen pistil interaction of the pollen grain and egg fusion decreases. In this way, what happens? The maturity period of the anther and the stigma be different. In this way, when the anther matures, at that moment, the stigma does not mature. Egg does not mature. Means if the anther matures, anther release the pollen grains, and if that is dusting over the stigma surface, the pollination will not take place. In this way, self-receptivity decreases, and the pollination and the self-pollination chances totally rejected, totally become non-synchronized. The second thing is that the plant produce unisexual flowers. If the plant is producing the unisexual flower, means if in the case of papaya and date palm, we have studied yesterday in the previous class, that in the papaya plant, one plant is either a staminate or one plant have the flower that will be pistillate. In this way, divorcee plants are created. In this way, self-pollination chances are decreased. Third is that the position of the pistil and stamen are different. If the pistil is slightly curved and a stamen is on the different position, then the pollen grains after getting mature, if dusted, that will not settle over the stigma because their position are on different directions. And third is that self-incompatibility. If the fourth is one the self-incompatibility. What is, if the pollen grain is dusting over the stigma, then the stigma will not identify the pollen grains of the same flower. In this way, this is called self-incompatibility, means that stigma surface will not allow the pollen grains to germinate over the surface of the stigma. After this, the next topic we have to study about the pollen pistil interaction. Pollen pistil interaction means how do pollen grains settle over the surface of a stigma, and how the pollen tube forms through the zygospore and that penetrate the surface of a stigma and reaches to the ovary and ovule and supply the male gamete to the egg inside the ovule. You already studied about this figure, but you can see the figure once. You can see here these are the pollen grains, and these pollen grains are dusted over the surface of a stigma, and these are making the pollen tubes. And these pollen tubes are germinating, and pollen tubes are taking the male gametes, and male gametes are carried to the ovule. Inside the ovule, egg is formed, female gamete is formed, and that female gamete gain this male gamete, and fertilization is facilitated. So this is the interaction. This figure is showing the interaction between the pollen grains and the pistil. So this term is used for the pollen pistil interaction, and this is the pre-fertilization events. You know well this pollen grains and this pistil interaction, how the germination taking place of the pollen tube, and that is carrying the male gametes, and that male gametes are served to the ovule and to the egg for the fertilization process, so that the zygote formation and further development can take place. So this type of activity which is taking place in the plant body, that is termed as pollen pistil interaction. The next term we have to study today, that is artificial hybridization. You know very well that artificial hybridization in the 1960s, if we are going, we know that green revolution was performed, and that was the revolution which brought the happiness in the huge population possessing our country. At that moment, we have the large population of India were below poverty line. They have even not the two times food for their survival. In this case, you can recall the scientists names of the scientists, that is the Norman E. Borlaug and M. S. Swaminathan. You know that between this hybridization is the process of cross between the two desired qualities, plants or crops, having the desired characters, having the desired traits of the high yielding varieties. Hybridization is the process of cross between two good qualities to obtain the superior crop, to obtain the superior hybrid. If I am identifying that if the particular variety of the plant, particular variety of any crop having the good quality, having the large productivity, having the semi-dwarf size, have the lodging resistivity, have the logging resistivity, have the desiccation resistivity, and a particular plant which is very good efficient to survive in the Indian climatic condition, have the fragrant and the large flowers, have the better pollination capacity, have the better fertilization capacity. In this way, we can use these two types of crops and make them hybridized. But we know that generally the plants are bisexual. If these plants are bisexual, then before you take the desired qualities, pollen grains, and dust over the surface of a stigma of the desired qualities crop, it may be that self pollination can be performed before the cross pollination, before the dusting of the desired pollen grain. Now we have to know that in this way artificial hybridization is done. In this way, what we do, we take all two types of plants and we remove the pollen grain of such type of plant. Suppose you are taking plant A and plant B, and plant A is bisexual and that performs self pollination. Then in this plant A, before the maturity, the pollen grains are removed and the stigma is covered by the butter paper. So that process of removal of the stamen or the anther from this bisexual flower is known as emasculation. After emasculation, as we know that pollen grains and different types of pollen grains float in the air, and that unwanted pollen grains can fall over the surface of a stigma. So we cover the surface of a stigma by the butter paper, and that process of covering the surface of a stigma by the butter paper to prevent the falling of the unwanted pollen grains is known as bagging. We will take the anther of the desired quality crop, and that will be dusted after opening the bag of the stigma, and we will dust over the surface of a stigma the desired pollen grain that is of human interest. And now again the butter paper used to cover the surface of a stigma to again prevent the unwanted pollen grain falling over the surface. The process of recovering of the stigma after the dusting of pollen grain of the desired quality, that is called rebagging. And after rebagging, these two desired qualities pollen grains and the egg will be pollinated, will be fertilized, and will be making the embryo and seeds, and seeds will have better quality character, will have the superior quality, and that will be fulfilling what the artificial hybridization purpose was earlier. Thanks.